When we're looking at determining if evolution is occurring, one of the major things that we do is try to quantify or measure if there is evolutionary change. And so we've just learned about some equations that are going to help us do that. We've got the allele equation that looks at the two alleles of a gene pool, the whole gene pool. And then we look at the whole types of individuals and the 3 different genotypes that the individuals can have. We keep in mind that the natural selection force is acting on the phenotype. So whatever it is that is allowing some to survive better than others, it's selecting the phenotype. However, which phenotypes reproduce do have an effect on which genotypes and therefore which alleles are passed on to the offspring. So we did some practice with some equations. We looked at being able to find the frequency of the genotypes, the frequency of the alleles going forward and backward. So now we look at what are the real applications of Hardy-Weinberg. One of them is just the truth that we can predict the occurrence of all alleles in a population. So when we look at a certain group of individuals, in that population's gene pool, which alleles are present in that gene pool. And we can compare populations across the world. And so we can see if the populations have a lot in common or if they're very different. And then we can begin to predict what caused those differences. The second thing that we can do is determine the percent of genotypical heterozygotes. So when we did genetics, we used to just say, well, if you can roll your tongue, you are dominant. I don't know if you're dominant, homozygous dominant, or if you're heterozygous, I just know that you're dominant. We still can't figure out individuals, but we can figure out the percentage. So we still can't look at an individual and tell if they're a heterozygote or a homozygous dominant. But we can tell the percentage of the population. And so what we can do there is we can make predictions about the chance that you are heterozygous or homozygous. And we can also really start to kind of break down our data, and this allows us to predict what the next generation will look like of the population. So before, when we were making predictions, we were just using 2 parents and making a prediction of the parents. But now we're able to actually look at the whole population and make a prediction for the population. So here's a really interesting set of data, and this is frequencies of the sickle cell allele. Now keep in mind that sickle cell is actually co-dominant trait. And this allele is going to be the allele for the misshapen red blood cell. So what we see when we look at the frequency of the allele is we see certain pockets where the frequency is significantly higher than in other areas. And if we were looking at a global map, we would actually see a very similar trend very close to the equator. It turns out that there is something called a heterozygote advantage. Heterozygotes, remember, are things like big B, little b, or sickle and normal. This one being a codominant, this being dominant and recessive. When we looked at the problem in the textbook of what happened if all the white cats died, if all the recessives died. So a really good example is if we look at this gene and we have sickle normal, we would also have normal, normal, we also have sickle, sickle. Sickle, sickle individuals are actually very sick because of sickle cell. And so they usually die or don't live long enough to reproduce. Normal, normal, turns out they're normal, but they are very likely to get malaria. So they have a high risk of malaria. And A-L-A-R-I-A, high risk of malaria if you're normal, normal, because malaria is caused by a parasitic unicellular eukaryote that lives in the red blood cells. So this is just one case of where if you are a heterozygote, you have an advantage. Because the heterozygotes have enough normal cells to not have sickle cell. But enough sickle cells that these parasites can't live inside of their bloodstream. And this is going to happen in a lot of other cases. This is really just the most famous one, the one you definitely want to know. In that, heterozygotes have this hidden source of variation. And this hidden source of variation is going to increase the health of the population. Meaning, if something comes along like malaria and we were all normal, then we would all have a much increased chance of dying. But the fact that we have this hidden variety, which can also produce something bad if we have SS, that hidden variation is going to increase the health of the overall population. And so that's one of the things that we can do with Hardy-Weinberg is we can actually look at, now that we can calculate the frequency of the alleles, we can actually compare different populations. We see that the highest rate of sickle cell anemia is in African populations around the equator, because that's where this particular mosquito that carries the malaria protus, Plasmodium falciparum, actually lives. So a long story to say that hidden variation helps with the health of a population. As we move on, we start to look at what else can we use Hardy-Weinberg for. The second thing that we use Hardy-Weinberg for is the idea that Hardy-Weinberg equilibrium assumes evolution is not occurring and that allele frequencies stay the same from generation to generation. So if my P is 0.7 and my Q is 0.3, if I look 10 generations later and my P is 0.69 and my Q is 0.31, that's not a significant change. Therefore, my allele frequencies are mostly staying the same. Therefore, evolution is not occurring. However, what if another 10 generations happen, and now my P is 0.4 and my Q is 0.6? That's a pretty drastic change, and so that means that something is happening. And so therefore, we start to question what is causing this change. And there are 5 main things that could cause the change, and you need to just memorize them, but you can use logic to understand. First, and so these conditions mean that evolution is not occurring. And so if these are untrue, so no evolution occurring, if true. So if no evolution is occurring, I can assume these are true. If I know these to be true, I should expect no evolution to occur. But if there is a change, that must mean that one of these is probably wrong. So it could be that I don't have a large population size. That means there's a small number of mates. If I have a small number of mates, we're going to see much more variation in the allele frequencies. When Hardy-Weinberg is at equilibrium, random mating is occurring versus sexual selection, which would cause changes in the allele frequency. Under normal circumstances, we don't have mutations. So if we're seeing some pretty drastic changes, there might be a mutagenic agent. Under normal circumstances, no genes are input from other sources versus immigration. Under normal circumstances, no selection is occurring versus if we have changes, selection might be occurring. So you want to be able to kind of memorize these almost forward and backward. If the circumstances are normal and there's no change, then these conditions are probably true. If I'm seeing changes, then one or more of these conditions is probably false. And so we have a really interesting generation here, generation 1, 2, and 3. Interestingly enough, this is actually an incomplete dominant trait. We can still use Hardy-Weinberg because there's still only 2 alleles, the red allele and the white allele, but we just see that when they're together they mix. And so if we look at this, we can actually see a pretty drastic change in the allele frequency. We can predict what is happening. We don't see any genes being input, but we do see the loss of certain phenotypes, which means we're also losing the alleles as well. This is pretty drastic to happen over just 2 extra generations. When we move on and we look at the last set of notes, we see that these are kind of the opposite of what we just saw up here. What this says is that under the following conditions, the Hardy-Weinberg equation will predict evolutionary change. So if there is mutations, I'll see evolutionary change. If there is gene flow, now I want to kind of help you out here with what gene flow means. This is or emigration. This is genes flowing in or out. This is directional. Non-random mating, we can make sense of that. Genetic drift. Drift is going to be random. So this is going to be like if I put a net into a pond and scoop up a bunch of fish, and I start a new fish tank with them. I didn't select them. I randomly chose who was going to survive and not. And so genetic drift is when there's usually some sort of random environmental change. And then we have natural selection, which we've already talked a lot about. So where you need to be at this point is you need to be able to kind of make these fit with the 5 terms above because they're basically opposites of each other. If these are happening, evolutionary change is happening. If those conditions up above are true, then no change should be happening. So they're really just a way to look at it. And when you are working with these 5, there are some great pictures in your textbook that you're going to kind of copy and draw, and you're writing a slogan for them. And so the main idea is, how do I describe gene flow? It's emma or immigration, genes flowing in or out in one or more directions. Or non-random meeting. That means to pick your mate, select your mate. And so what we start to see is that these are the things that are probably happening if I see changes in a population. And so you want to make sure that you can kind of memorize these. They're logical as well, but you just have to practice them so they get into your memory as reasons that evolution could be occurring. And that's it.